record. All right. And we are recording as before. Okay, so let's get started. Thanks for coming back. I wanted to start by going over some things that came up over the last week in terms of issues folks were running into, with the last material. And I realized last week was pretty tough because we weren't actually able to go through and do a lot of the stuff live because some of those computations take a while to do. Good on you for working through that if you were able to do that. I think the thing that was the most problematic is getting things loaded up properly, reading the process data that comes out of CellRanger, reading that off of the disk and getting that into R. And you always have to do some sort of fiddling with the file paths in order to make those correct and specific for your computer system. So there's no way for me to sort of prepackage that and tell you how it's going to look. So let me just show you more clearly how to do that. And so this is the command that we used in the class last week. And what this is doing here, this is reading a CSV file out of a package. All right. So this package is something that, gets installed when you load up the tools, Blazor tools package. So this data is just for the vignette, for the example. But what you want to do is you want to make that value read, something, that value be where the file is on your computer. Okay, so let me show you how you can do something like that. So this is the command that we ran before. And so that's the table that we were using for the example last week. And what I'm doing here with this command is I'm just writing it out to a place on my on my drive. Now that gave an error because I actually already did it and I already have the file open. So ignore the error, but here is that file now written out into a CSV file that I've opened up in just regular old Excel. And this is where you're going to take the opportunity to customize it. And in particular, what you would want to do is you want to customize this file path here. And you would make that specific for wherever you had those data on your system, okay? And one way you can do it, this is another source of confusion here, is these file paths obviously are pointing directly to a .tar.gz file. That's a compressed archive file with with 3 other files inside of it. And that is actually the file that's going to be used by the main program inside the script to load up your data into a cell dataset object. So that's what it's actually looking for. But you might want to edit it depending upon how the data is stored on your system. You might want to edit it to go for not the file itself, but the parental folder. And this has to do with what version of CellRanger you're using, because sometimes, those files are going to be a little bit hard to find, or they might be buried someplace that's different and is difficult to point to, whereas it might be easier to point to a higher folder. And that's something you're going to have to play around with, and I can't give you any more better instructions than that at this point. But you should be able to just walk through this code here. And this is the code from the load process file that we used last time, and see exactly what it's doing. And so because these are all concepts that we've gone over and hopefully you're going to become familiar with if you're not already. And so just to walk you through this, what this function is doing is it's mapping through this column here, the sample column. The sample column holds only 2 values, the 2 names of the samples, all right, because there's 2 samples. And what it's going to do is it's going to apply those to a function here, all right? This function is going to, so basically each value in sample is going to be transported into this value x. It's also going to read in another value here, which is the whole configuration file, which is everything else that you've included, on this file here that you're using. Okay, so once you've read in your configuration file, that's going to get sort of transported into this function as well. And what the first step is doing here is it's taking and it's basically stripping out the correct line from this file. So it's getting all the data from each row on this table. Okay, that's what this conf filtered is. Okay, and then the next chunk here is a search function, basically. It's basically going through the parental directory and sorting down and finding wherever it's going to be able to find this exact file here, filtered featured barcode matrix.tar.gz. So that has to be present somewhere in the directory of your data that's holding your data. Okay, and so what's that's going to do is it's going to pull out that exact file. So that's what you need. And then it's going to put that into this function here, which actually takes those data and loads it into the cell dataset object. The rest of this table that you've made becomes the sample metadata. Okay, so like all the other things you already know about the sample that you want to put into your data object, will be in this table. So if, this can be, dozens of columns or hundreds of columns, however many columns you want, it will, that will be attached to every sample in your data set. Okay, usually you don't want the file path in there. Okay, in this case, it says tar gz path. So you might say, okay, well, let's get rid of, see tar gz path. Because, you definitely don't need that in your data. That's not helpful. Tar gz path. Okay. So yeah, so then the other thing you don't need in there is the sample name, because that actually, it's a little bit confusing, but later on will cause problems if you leave that in there. But you do want to put that as the name of that object. So that's where the sample name gets attached to your data on that one. Yeah, so that's how that works. There's now a variation or a variant of this that will work for not just the tar gz, but also the H5 files. That's in the latest version of the Blazor Tools package if you want to update that. That's if, whatever you're doing, whatever pipeline you're running on Cell Ranger produces just an H5 file. And like sometimes that will happen as well. So I realize that's a pretty confusing and, sort of finicky type of thing to do. It's just going to take a little bit of patience, and definitely you have to read the error messages. So if you get an error, it's usually going to tell you what the problem was. The other thing I realized as I was going through this, I didn't show you, is how to actually, yeah, so sorry. So in any event, I also wanted to show this. So in order to, once you've edited that CSV file, all you're going to do is basically just read it in with a very similar command as this. So, we can do that, and then we can read that, right? And then we'll make this the file path here, like so. And that should read it in, because that file, I just wrote it there. Okay, so it read it back in. So that's how it would normally look on your system. You just have to make sure it's pointing to the right file. Okay, good. So so that's that. And what I want to do now is go through really more the on the more of the biological side. How are you going to analyze your single cell RNA-seq data? In other words, how do you think about it? How do you approach it? How do you determine, like what's meaningful? And how do you communicate that to, in your thesis or in your manuscript? Okay, so And I think the hard part here is that we have heterogeneity, right? So yesterday we sort of left off with defining some clusters of cells using different algorithms, and if you have questions about that, you can put those up in the chat. But really, a cluster is a classification of cells determining what groups of cells are similar to each other. And you can use various resolutions and various algorithms to do that. But in the end, you come up with cell clusters. And these cell clusters will contain cells from different samples, and therefore they'll contain cells from different experimental groups if you're doing some sort of an experiment, or it will come from different patients if you're characterizing a group of patients, and each of these patients will have different variables that describe them, like age, disease, gender, characteristics of the disease. So you can, really, use any arbitrary variable uh, um, as another classifier for your cells. And so the difficulty becomes in determining, does my variable of interest, say experimental group versus control group, does that, how does that explain the heterogeneity of the different cells? Okay, so if I'm looking at two cells, and here's the problem: if I'm looking at two cells or two groups of cells that are put together in UMAP space, in other words, they're right next to each other in the XY dimensions on a UMAP plot, by that very assumption, those two cells are similar and there's not much difference between them. So you have to find a different group of cells to compare those to. And so what group of cells do you compare them to? And so that becomes a major challenge when you're trying to determine, how the different variables in my experiment affect the different clusters, and how do I do a differential gene expression between different clusters and different variables? So it becomes too complicated. And so the way that you want to address that, and this is a very effective method, although it's a little bit counterintuitive, and a little bit different than saying, I want to just compare my experimental group and my control group. What you want to do is define all of the clusters of interest and characterize them. And you can use a number of methods to do that. You can do the top specific genes for those clusters. You can map to a reference data set. You can do differential gene expression between clusters. There's any number of ways that you can fully characterize a cluster. And then in a second step, you want to show differential representation between those cell clusters according to your experimental class. So say you've defined a B cell group, a T cell group, and a monocyte group, which we're going to go through today. What you want to do is characterize those and say, here's all of the genes and all of the gene expression and maybe cell mapping identities that characterize the B cell group and the T cell group and the NK cell group, et cetera, et cetera. And then say, okay, well, in sample one, I have a certain number of cells in each of these clusters, sample two, a certain number of cells. And then according to how your samples fall into those experimental classes, then that tells you how the heterogeneity has changed between your samples according to experimental class. So this very nicely handles the problem of what do you compare each of your cells, each of your classes to? How do you pick the right cells to compare to? And it also handles the problem of, if I compare too narrow, I'm not going to find any differences between two cells that are lying close together or two groups of cells that are lying close together. So I think as you get your own data and get some more, some more data sets, and hopefully by the end of the day, we'll have an example for you, you'll see how this really makes a lot of sense. Okay, so so the first thing that you want to do when you're sitting down to work on this is you want to visualize your data. And this is going to help you explore it and understand a little bit about, what your samples look like in terms of their transcriptional state. That's really what we're measuring here. And by this I mean the UMAP dimensions. These are going to be a lot of UMAP plots. So your goal here with this visualization is to show global transcriptional state in individual cells. You show how cells cluster according to their transcriptional state. So how you've classified cells, from the whole experiment together. You're going to want to show the distribution of sample level cell metadata according to transcriptional state. So what does that mean? Basically, where each sample lies in the merged UMAP plot, where each experimental group, where each sort of patient characteristic variable lies. And that will give you an idea of how the transcriptional state varies between your groups. To show expression of single genes or groups of genes in groups of cells. So, that's going to help you characterize each of those cells. If I say it's a B cell, it should express, CD20, that sort of thing. And so hopefully, you're going to be able to build on the principles of data visualization that we've started to introduce in order to do this. So, you want to make sure that you choose the right plot and the right colors and the right sort of parameters to make a fair representation of your data that doesn't confuse you or mislead you nor your reader. So let's just talk about the where these plots are coming from. Okay, so all these plots are going to be essentially stripped out of data that is within the cell data set object. Okay, if you're going to make a UMAP plot, very very simply, this is going to come, the X and Y dimensions for each cell are going to come from the UMAP dimensions that are built in or lying within the cell data set object. Okay, and you can pull those out with that command there. That's sometimes, but usually not a useful command that you need to run. But when we're going to run some plotting functions, that's essentially how it's going to get those XY coordinates. So X and Y is one or two dimensions or two aesthetics that you can use. Obviously color is another one that's very straightforward and easy for people to understand. So you can pick any other value that you want to represent by color. And this can be either discrete values like some sample level data or some clustering data. And each of those values in that variable can be represented by a color. You can also pick a continuous variable. So let's say some expression value for a gene or some pseudo time coordinate if you've done that. And that can be on a color scale as well. All right, so usually have X, Y, and color that you can use. For cell metadata, it's usually going to be a discrete variable that you're visualizing. Expression data is also going to be shown in color. It's going to be the functions that we use that I'm going to show you are going to produce normalized expression values. And this is going to be plotted on a color scale. There's a lot of ways to change that if you feel like it's being misleading or if it's not showing what you think it should be showing. For example, if you try and show two genes next to each other, it might change the scale to fit one but not the other, and so you might have to do some workarounds to show each of the cells in their own scale. So that's a little bit of a complicated thing, but once you understand that where these data are coming from, it becomes less of a challenge to go in and tweak those things if that's what you need to do. You can also plot other data in these formats as well. So if you're doing scRNAseq, or if you're doing, if you've made some pseudotime calculations, all these things can live within the cell data sets. They get pulled out and they can be displayed as a color, or as a an XY coordinate. All right, so most of the plots that we're going to use are going to be ggplots. These are simply the most flexible and best looking plots that you can generate in R. And so anything that you do that we're going to show today is going to return a ggplot. So as we showed before, you can always add layers onto ggplots. So if the thing is coming out and you want to change something after the fact, like like put it on a different color scale to match something else that you're doing in your paper, that's like really easy to do. All you have to do is just take that plot and then add the scale to it afterwards. And you can go back and look when we talked about ggplots how to do that. Even though it's complex, really, and maybe there's a lot of points on it, it's really no different than any other ggplot in terms of how that figure is constructed. Okay, one of the things that we're not going to talk about are heat maps. There's a lot of heat map functions out there that you'll find, and I'm happy to point you in a direction if you feel like you need to do that. I think most of the time you're not going to need to show a heat map. There are better ways to communicate that sort of data, which I'm going to show you in a little bit. But yeah, if you need to show something like that with a heat map, I can point you in the right direction. But they can be a little bit complex to construct in a professional looking way. Okay, so the functions that we're going to go through, I'm going to do these right now, that I'm going to show you how to use, come from my Blazor Tools package. I find these, basically cover, 95% of the things that I want to do for, any sort of paper. If I want to show cell metadata, which is any sort of cluster or sample data that we know about any individual cell, I'm going to use the BB var UMAP function. Var stands for just variable. So that's what that means. And if I want to show gene expression in a UMAP plot, I'm going to use the BB gene UMAP function. All right. And then once we go through some of those, I'm going to show you this gene bubbles, which I think is the alternative that you want to use. A lot of times if you think you want to show a heat map, I think often gene bubbles are a better approach. Okay, so let's go to R now. And anytime I'm sitting down to explore data, the first thing I'm going to do is I'm going to want to know what's in my data object. And usually I start with the cell metadata because that's the most where the most sort of interesting stuff is. And so this function right here, cell meta, that is a wrapper. I mean, it's not complicated. All it does is it gets the cell metadata out of the CDS object, and it presents it as a tibble, which is the most useful form of a data frame in R. And usually it looks like that, which is sort of helpful. So you can see there's 1160 rows in this table, meaning there's 1160 cells in the data set. All right. And if I want to just sort of summarize it in a way that's a little bit easier to use, to read, I can use this glimpse function. That just shows me all of the columns here. And I can pick any one of these that I want to visualize. Okay, so let's say I want to see sample. All right, well, there's pretty much always going to be a sample variable in there, so that's an easy one. So if I run the var UMAP function on the CDS, I get this. Okay, so it is a pretty typical UMAP plot, X, Y, and here each color is indicating what sample this came from. All right, so that's pretty straightforward and useful. And you can see here in these data, the samples pretty much overlap in UMAP space. So they're very similar, which, by the way, was the point that the people that made that data set were trying to get across, is that their two devices produce similar results. Okay, so that's good. So I think, towards the end of last week, we went through how to make clusters, okay? And it's pretty, it's pretty automated. I usually don't mess around with it. I pick, I calculate three different clusters, three different resolutions of clusters, starting from the most coarse to the most fine. We have the partitions, the Leiden clusters, and the Louvain clusters. And so we just sort of arbitrarily, run those, or we run those sort of automatically, and then calculate the top specific genes, the top 50 or so specific genes for each of those clusters. Okay, so let's see what those look like. You can see here, if we look at the partition, there's only one partition. So, that was so coarse it didn't really identify any heterogeneity at all. All right, so that's not going to be too useful. All right, so this one here identified three clusters. All right, so that's helpful. And the Louvain algorithm identified 18. Wow. Okay, so that's a lot. And maybe that's not a great place to start if we're not, if we don't, really know what we're dealing with yet. So usually I would pick something that looks like this in order to start to drill our way down into what's going on. And as you can see here, if I run this here, this top markers, this is just a table of all of the different top genes from all of these clusters put together in a giant table. And that comes out automatically from the processing script that we went through yesterday. Okay, so if I go back, I'm just going to quickly bring up the end of the script. We did not get to this last week. One of the challenges is, what's cluster one? I mean, that's not great. We want to figure out what those cells actually are. And, you can scroll through the top genes, and that's helpful. But there are, and I think a lot of you will be interested in using automated methods in order to map those cell identities to something that's been published previously. By now, there's a ton of single cell data out there, and you might be able to find a reference data set that's applicable to your samples that you can map to, and then say, okay, well, I have some probability that my cell is a memory T cell, a CD8 memory T cell, because it maps to this other reference data set, which is well characterized and published, et cetera, et cetera. So, we have a number of functions that enable us to do that. That's not the hard part. The hard part is actually getting those data sets in a way, in a format that's going to be, helpful and useful for you. That requires some experience to be able to do that. And the reason is you're not like, people don't publish their process data usually in order to make it easy for you to work with. So you usually have to go back and get, the raw data and make your own data sets. In any event, there is one published reference file that is useful, okay? And this is from Human PBMC, and this is produced by the Seurat people. And you can download this file from the Seurat website, and I think you just have to Google that and you could find it. But essentially, this comes from, it's a pretty large file. It's, I don't know, it's a couple of gigs in size. You download that file, and then you run this command here. All right. So Seurat anno, okay, annotate, and you're going to run that on the CDS that you're using. And the reference is simply that file that you download. All right. So you are not going to have that exact file there, or that package, because that package isn't published because it's so big. But if you download that file, put it on your computer, and then point this reference argument to that file, it will run it for you, and it will annotate your data with the identities from the Seurat data. And these are very good, well annotated, very descriptive and helpful annotations. Okay, so that's good. So that is how you do that. Now, let's say you're working with mouse bone marrow. Well, there's like no great reference, and so you're going to have to work a little bit to do that. And if you're working on a project like that and that's really of interest, you can definitely reach out to me, and I'm happy to help you. Okay, so what do you get out of all of that once you've done that label transfer or those cell annotations? Well, what that function is going to do is it's going to put some columns in your cell metadata for you. All right, it's going to put 4 columns in there. All right, it's going to put the Seurat dimensions and the Seurat cell type L1 and the Seurat cell type L2. All right, so think of Seurat cell type L1 as a more coarse designation of the cells, like T cells, B cells, and L2 is a more specific or a finer resolution like classification. So here, CD4 T central memory. All right, so you'll get those 4 columns tacked onto your data. All right, and so what does it look like? So we can plot that. And so that looks weird, right? Well, you can see here, this is Seurat dim1, Seurat dim2. What I've done is I've shown you these dimensions as opposed to the dimensions that we calculated in our data or in the vignette data. And so what has happened is each cell has been sort of put into think of it as like a sieve or a filter or whatever, and has been sorted out into the spot, the dimension reduction space where it belongs the best within the Seurat reference. Okay, so you're seeing here a sort of a shadow of the Seurat reference data set. Okay, so, and then wherever that lands, that becomes the label for that cell. All right, so what we can do is, and actually what we should typically do is we should show the data in the context of our own dimension reduction of the own sort of transcriptional state in our data, not the best fit for the Seurat data. Right, so we want to show it back in the in our sort of dimensional reduction, and this is the sort of thing you should get. All right, so these shapes should look familiar, but you can see we've now transferred these Seurat cell type L1 labels, and all the labels that were attached to each cell are now carried into this figure here. All right, and to show you the difference between L1 and L2, we can plot that one there. Okay, so now we have a very sort of intimidating view here, but still, high resolution cell assignments from the Seurat reference. Okay, so based on what we want to do, we can pick one of those and sort of work from there. Okay, so let's go back here and let's say, all right, well, if I look at my data, I'm, I don't want to get too like in the weeds right off the bat. So I want to work with the Leiden clusters. It's pretty simple. We only have 3 clusters we have to deal with. So let's start there. And then, if that's working, we can get more specific later on. Now the problem is, as I've shown you, if we look at this, this L1 designation, you can see here that you've got this is going to be like the T cell and NK cell cluster here, I think. But you've got some green cells here and some, orange cells there, and they're right, sort of intermingled. And there's a green cell, sort of like walking its way into the orange cells. And, that doesn't entirely make sense because, those cells are probably very similar. So, and same thing over here. You have some green cells over here that maybe, why are they detached from that cluster over there? So a lot of this, causes some problems because, we would want our clusters to be, sort of more cohesive and more consistent. And if there is a transition from one cell type to another within a cluster, we would like that to be a clear demarcation rather than a sort of blending of those cell types into each other. So we have to figure out a way to handle that problem. In other words, we're trying to figure out how to convert these individual cell type assignments that came from the reference into a more cohesive assignment for a whole cluster, okay? Because the cluster is representative of our data more than the assignment from another reference data set is. Okay, so how do we do that? Well, we can just do some very simple data manipulations to group those individual cell assignments according to the cluster that they come from in our data. And that's what this does here. And this just puts together a really small table, and then we can plot this table. And what this is showing us is that here, each column is one of those lighting clusters that we defined in our data. And this is a summary of all of the cell type annotations that came from the reference data set. And you can see here, one has got some blue, green, yellow, and pink. All right, so what is that? That's CD4T, CD8T, NK, and other T cells. So that's like basically all like T and NK cells. Okay, this is monocytes and DCs, and this one is pretty much all B cells. All right, well, I mean, definitely those are pretty clear. This one you could say, well, that's a pretty heterogeneous population, and so we could, have to figure out, do we want to deal with all that heterogeneity, or do we want to sort of lump it together and say, oh, well, this is like the T and NK cluster. So that's the decision you can make depending upon your own data. For the purposes today, what we're going to do is we're just going to call those T and NK. We're going to call 2 DC and monocyte, and 3 is going to be B cells. Okay, so now when you do something like this, you're going to want to, put that figure into your dissertation or your paper and say, okay, well, we're calling these things a certain name. We've assigned a name to it. And here's a justification for that. And so this is a pretty clear justification for those labels that we've put on there, I think. All right, so now we can, what we've done here is we've added that as a new column on our data. Okay, and then we can just visualize that now. So we have the same clusters, lighting clusters as before, but now we've put like a new and actually helpful name on there. All right, so T and NK, DC and monocytes, and the B cells up there. All right, and if I want to like get rid of the legend and like move the thing over, we can do this. We can do overwrite labels equals true. Okay, and that will put the labels like on top of the plot so we can see that. All right, good. So, all right, so that's good. Now, there's a lot of variations on this theme that you can do, and a lot of this is sort of like aesthetic choice, and picking something that communicates well and maybe provides a consistent theme from figure 1, all the different panels in figure 1, figure 2, etc. Connecting certain themes across figures is actually an important way to effectively communicate your data. And so these functions allow you to do that. You can set up certain palettes for different cell type classifications or sample identities, that sort of thing. So, I'll just demonstrate a few things here, and you can play with this. But, one nice thing is there, I want to just show the T and NK cells. So you can use this parameter here, value to highlight. I'm going to pick that particular value from the Leiden assignment column to highlight. Okay, that's cool. I don't like the color, so I can like make it green if I want to do that. So that's good. So there's a lot of ways that we can work with that. I'm going to move that guy down there. Sorry, got that. All right, so I'm going to go back up. So yeah, definitely, picking an aesthetic thing that works to communicate your data is something that you should be should be doing. All right, so I don't see any questions, so we're going to keep moving on. So, all right, so if you remember back in the beginning, I said, okay, you want to characterize your cells, and let me just pull that up here. Okay, and so we want to characterize. We talked about top specific gene. We've talked about cell identities, and we want to now like sort of validate those identities in some way. So, all right, if it's a T cell, it should express like T cell genes, right? So, okay, so CD3 is a T cell gene. So if we want to show expression in a UMAP format, we can run the gene UMAP function, all right? So that's pretty clear. So CD3 only lighting up really in these cells here, and it shows a color scale for expression. Okay, great. We can also show like some very typical genes for monocytes here, CD14, CD19 for B cells, and CD3 here. Okay, so that pretty well, establishes, at least to the level of 3 genes, that's what those clusters really are. All right. We can also show aggregated genes. All right. So maybe you want to show like a set of genes. It could be an arbitrary set. It can also be a gene module. And we calculated gene modules last week. What gene modules are? They are groups of genes that vary in a similar way from cell to cell to cell. All right. And they're not calculated based on any preconceived notion of what those cells are. They just say, this set of genes goes up together across, all 1,160 cells. And they go up and down together. And this other set of genes goes up and down together in a different way. And this other, set of genes goes up and down in yet a different way. And so these are called modules. And we can look at the different modules because they are stored in the row metadata of the CDS, okay? And here they are. So here's module. So some genes, they don't get a module. They're either like housekeeping genes or they're not expressed. But that gene there, whatever it is in module 1. That gene there, whatever it is in module 2. So, okay, so that's cool. And so what we want to do is we want to generate a tibble. Okay, I'll show you what this is here. All right, this is a 2-column tibble. First column is the gene ID, which should be a unique ID. In this case, the ensemble gene ID. And the other one is the grouping variable, in this case, gene module that we want to use. And in this case, we're only looking at module 1. We could put all the modules in there, and in that case, we would show all the modules. But in this case, we want to show just module 1. So if I run that now, if we run that, we can get the aggregated genes, and we can show that module 1 lights up very clearly in the monocyte and DC cells. So that's like a set of genes. If you looked at them, they would be all the monocyte genes, and you can then take that and plug that into like IPA or something, and it would tell you, what those genes are. All right, so that's a pretty another good way to validate these cell clusters that you've identified. Okay, so so this is good. There are some problems with this, however. One problem is that oftentimes showing the per cell expression of genes can be a little bit deceptive, and this can happen when you're plotting, say, tens or hundreds of thousands of cells, and you have a lot of cells that don't express a marker, but some that do, a few that do. And so how do you visualize that properly without unfairly biasing your eye? And so that can be a challenge. And I would encourage you to look critically at these things and plot things in a couple of different ways in order to validate for your own self that you're looking at the right stuff. And so I think oftentimes a more fair way to visualize this, although it's not as visually impressive sometimes, is with what I call a gene bubble plot. And in this case, what I'm going to show you is going to incorporate a fourth dimension of data. The first two are going to be a cell group and a gene. The third dimension is going to be per cell expression. And the fourth dimension is how many cells in a cluster that you're showing express that gene. Okay. And so this is going to handle the problem of dropout or cells not expressing any gene of interest. All right. So I'm going to run this one. And this is going to return a ggplot here. And as you can see here, there's 4 dimensions. On the X, we have the cell cluster that we've identified. On the Y, we have the 3 genes that we're interested in showing. Color is showing per cell expression, and the size of the spot is showing the proportion or fraction from 0 to 1 of cells in that cluster that are expressing that gene. And so this very clearly tells you, how specific and how strong each of these expression of these genes is. And so this is the way I would encourage you to show and validate your clusters, your cluster assignments, let's say, in the sort of the final analysis. I think the UMAPs are good for exploring the data, and they're good for you to get an understanding of how gene expression is heterogeneous across your data. But I think this is really a more reliable way to show how your markers are expressed in different cell clusters. Okay, so one thing that's done here is that the expression of each gene is scaled within itself. So in other words, the scale is going from, like, say, some negative to positive value, which is different for each gene. And that's because some genes are, like, just by their very nature, are expressed more highly than others. So we can turn that off by using this argument here. The scale expression equals false, and you can see, well, yeah, CD19, it's still expressed in more cells, but it's much more dim on an absolute basis. So that's something you're going to want to play with. And usually I keep the expression scale. That's usually a better way to show it. You can change the order of the genes and order of the cells by using these parameters here. So gene ordering and group ordering. Normally, by default, they're put in a clustering order. In other words, they're put close together in a hierarchical clustering algorithm for 3 genes and 3 cell groups. It's sort of random, but if you want to arrange it in a certain way, you just make the gene ordering as supplied and the group ordering as supplied, and then you just make the cell groups or the gene groups the way you want them to look. Okay, so that's visualizations. Now I wanted to talk about differential gene expression, because you may want to say, okay, well, so prove to me, these are, B cells, right? And then you say, all right, well, I can show you every gene, the differential expression, between 2 groups of cells. And so, all right, well, that's doable. Well, if you get to a complicated data set, there might be dozens of groups of cells that you don't know what they are. You want to show how these, cell group 10 differs from cell group 15, for example. And that's really, I think, we're getting to the point where you guys are going to want to know how to do this and not have to farm this out to a dedicated bioinformatician, because the number of comparisons that you're going to want to do is going to, once you start to get into a larger, more heterogeneous data set, is going to be growing, and it's going to get really large. So it's really important for you guys to be able to do these differential gene expression calculations on your own. And you get a table of genes out of it, and you can do whatever you want with those data. So, but the question is, how do you set that up, and how do you do it in a way that accounts for potentially confounder variables, and that sort of thing. So what I want to show you is a function that I've built, which is based on the core function is, I'm going to scroll down here, is based on the DESeq2 package. So this is a standard tool that we use in bulk RNA-seq analysis to determine differential expression of each gene between a number of samples. All right. And so the approach that we're going to take is we are going to make our own samples. Okay. And we can do that because we have all the tools, we have all the counts from all the cells, and all we have to do is select the cells that we want to use. In other words, we bioinformatically sort them. We can generate an aggregated count matrix from those groups of cells. Okay, and then we can just feed that into DESeq2, and we'll get a list of genes out with fold change and p-values and q-values and that sort of thing, and then we're good to go. What are the things that you need to know and need to do here? So this approach will consider variability in gene expression between replicates. So you have to have some form of replicates in your data. Now, we all know what true experimental replicates are. So if I have, one mouse versus one mouse, there's no biological replicates there. You have one control, one experimental. So that can be a challenge. And, if you're doing a good experiment, you might want to do 3 versus 3. That might be a better approach. But in fact, we can actually double the number of replicates we have for this assay, because we can consider pseudoreplicates. So what is a pseudoreplicate? Well, it's a subset of cells from a biologically distinct sample. So if we think about our data that we're working with now, we have 2 samples, so no replicates. But what we can do is say, all right, well, within the B cell cluster, I have 2 replicates, right? I have the chromium 1 and chromium 2. So that's n equals 2 there. Within the monocyte cluster, I have 2 replicates. That's from the chromium machine and the chromium X machine. So I have 2 versus 2. So I actually do have, what I call pseudo replicates. So they're unique subsets of cells from biologically distinct samples. Okay. And so that essentially doubles your number of replicates that you can use for this assay, because again, we're not comparing in this assay, we're not comparing group A versus group B. We're comparing cluster A versus cluster B, or the B cells versus the monocytes. And so that's I think where the heart of this whole analysis paradigm comes into play. And so once you do that, then you can then you can later on show differential representation of your experimental groups between those clusters or within those clusters. All right, so let me show you how that's done. And then I think we're going to wrap it up for today, and we'll pick up next week. Okay, so very easy to get these, in this case, I'm calling it experimental design, but you can just think of it as the pseudo samples, all right, or pseudo replicates. So you take your cell metadata from your cell data set, you're going to group by biological sample. So that's one component that goes into a pseudo replicate, right? Biological sample. And the other component that goes in there is the cluster, all right? And it's the cluster that we're interested in comparing. So that is the other thing that you're going to group by. And then you summarize, and what you get is a table like this. So this is exactly what I was saying. So within this variable, let's say B, we have 2 replicates, the chromium X replicate and the chromium controller replicate, all right? And so those become our pseudoreplicates for the B cells, and these are our pseudoreplicates for the DC and monocells, et cetera, et cetera. So that's our experimental design, or the pseudo samples, let's say. And so then all you have to do is you have to feed this into this function here. This it's pseudo bulk multifactorial. Pseudo bulk because we're doing a pseudo bulk RNA-seq on these pseudo samples, all right? And multifactorial because, you can put in other variables to account for confounders, which I'm not going to go through today. This runs very, easily. You put in the cell data set that you're working with. You put in this experimental design table, which is this thing we just built here. You put in the design formula. So what this is, this sort of tilde thing. It can be a little bit confusing, but really what this translates to is show me differential expression based on Leiden assignment. Okay. So what it's going to do, it's going to, figure out how expression of every gene that we've detected varies between all of the possible values of Leiden assignment, right? So there's 3 possible values there, T, NK, DC mono, and B. I'm going to figure out how that all varies for each one of them, okay? And then it's going to return a result. And the result is going to be a pairwise result. So you can only show, group A versus group B, but this is going to factor out all the genes for the B cells. So this is really only looking at the T and NK versus the DC and monocytes. All right. So that's like sort of an approach that is more specific for the for these data. And what this is going to do is show me the differential expression, again, based on Leiden assignment with the T and NK genes being positive log fold change and the DC monocytes being negative log fold change. Okay, so I can run that now. This takes just a couple seconds to run. And there we go. So what this does is it returns a bunch of stuff. All right, some figures and that sort of thing. But the main thing here is the header. So it's telling you the mean of the normalized counts for all samples, and it's going to tell you Leiden assignment T and NK versus DC monocyte, and it's going to return in the result element all the stuff that we want to see. Okay, so the genes, the whole table of genes, the log 2 fold change, the p-value, the p-adjusted value. All right, and so let's just look at the result here. And you can see you've got this table here, CD3, other things, all that you would expect to be positive in the T and NK cells. Okay. And yeah, so let's leave it there for today. You should, if you're able to get the vignette loaded up, you should be able to run through all these things on your own. I would encourage you to do that. It's going to run pretty quickly. And then you'll be, sort of on your way to working with your own data next. All right. Well, thanks everyone for joining. This is going to be up on GitHub here in a few minutes. And then you can look through this script on your own as well. All right. Have a good week. See you next time.